Thank you very much. It's a great pleasure to be here. Thank you for inviting me. I will review in my talk the STAT proteins. You've heard a lot about JAKs. Now, the STAT proteins are just downstream of the JAK protein. So we have here a receptor with its ligand activating JAKs. And the simpler version is that there are two downstream signaling pathways. One is mediated by these so-called STATs. And that stands for signal transducers and activators of transcription. These are phosphorylated, move into the nucleus, and directly affect transcription. And then also the signaling branches out into the more general pathways, such as the RAS-MAP kinase, etc. So we'll focus now on these STAT proteins, which are downstream of the JAK signaling pathway. And we'll consider primarily three members of the STAT family: STAT1, STAT3, and STAT5. And I will focus mainly on two cytokine receptors, EPO receptor with its ligand, and the thrombopoietin receptor MPL and its ligand. And so the consensus is that STAT5 is an important mediator of signals generated by these two receptors. And the JAK2, the mutated JAK2, amplifies the signals coming from the EPO receptor and from the TPO receptor, such as to increase the blood in peripheral blood. And so there have been two publications that define the role of STAT5. So the question is, what happens if you now eliminate selectively STAT5 using a mouse knockout? The first was, and the prediction is that you will normalize red cell and megakaryocyte platelet in these mice. So the first is by Rick van Etten's group. It was a retroviral model where he introduced the V617F into the bone marrow of these mice, transplanted, and then selectively deleted STAT5. And you can see that the hemoglobin, hematocrit normalized, except for an outlier here. Also the white blood counts were affected, and the spleen size normalized. So clearly in this model, the phenotype of V617F-driven MPN was dependent on the presence of STAT5. Here in a mouse knock-in model, the group of Ghulam Mohi did the same experiment and found the same result. So we have here the mutant mice, and when you delete STAT5, then hemoglobin normalizes, the platelets go down, and also the neutrophils. And you can see here the effect also on the spleen size here with V617F in presence of STAT5 and in absence, it normalizes. So clearly, STAT5 is an essential component, and as you know, you could probably deduce STAT5 is a potential target for therapy because interfering with STAT5 function will normalize the blood counts. STAT1 has been studied in several systems, and there have been data from John Crispino's lab showing that STAT5 is actually important for megakaryopoiesis already in 2007, and then Tony Green's group in 2010 showed that in samples from patients, STAT1 activity is upregulated when the disease is shifted towards a more ET phenotype. So the question then what we ask is what's going to happen if we delete STAT5 in mice, how it will affect the phenotype. The prediction is that the red cell mass should increase because in this system STAT1 has normally a negative effect, whereas the megakaryocytes are predicted to be inhibited, so fewer platelets would be the prediction. And this is what we found. So here are the control mice. Here are the mice with V617F alone, and if you knock out the STAT1, then hemoglobin goes up and the platelets go down. Not totally to normal levels, they're still elevated, but they are lower than V617F that is acting in the presence of STAT1. The spleen weight had in this model, no effect. So deleting STAT1 had no effect on spleen levels. And this is just to show that the knockout was complete. It translated into slightly less myelofibrosis. So in the presence of STAT1, we have myelofibrosis starting around 16 weeks or so in these mice. When STAT1 is absent, then we have a shift towards more mice in grade 0 and a few in grade 1, 2 myelofibrosis. The deletion of STAT1, as predicted, will affect the expression of target genes that are downstream of STAT1. And interestingly, we found that in patients with PV, ET, and PMF, the base levels of gamma interferon were slightly higher than the controls, and they were highest in the ET. Why is this potentially interesting? It's potentially interesting because the STAT1 is not directly targeted by the mutant JAK2. Instead, from the EPO receptor or TPO receptor, instead it is stimulated mainly from the gamma interferon receptor, which signals through a dimer of JAK1 and JAK2. So the mutant JAK2 then is sort of predicted to amplify the signals coming from interferon gamma, and stimulating STAT1 would then shift the phenotype slightly towards a more ET phenotype. So the elevated gamma interferon levels in the ET would sort of fit into this model and would have the tendency to increase the platelets at the expense of red cells. So then STAT3, that's another STAT member that is directly phosphorylated by JAK2. And this was, for a long time, considered a good candidate for intervention. STAT3 is essentially in many tumors, many solid tumors. So the question was, what's going to happen when we eliminate STAT3 in the context of V617F MPN? Now, the prediction or the expectation was that it would have would decrease erythropoiesis and would increase platelets. We did not see an effect on hemoglobin, so the effect on erythropoiesis was not prominent or not visible, but we did see an increase in the platelet levels. The blue curve is the one where STAT3 is missing, whereas the red curve is the one where V617F is acting in the presence of STAT3. In neutrophils, it had a rather lowering effect. This did translate a minor decrease in the spleen, but the survival actually, in the absence of STAT3, was actually worse than in the presence of STAT3. And this is just to show that the knockout was effective. In the histology, as expected, if we increase the platelet levels, this is sort of the baseline myelofibrosis that we see in these mice. And with the increased platelet counts in the STAT3 knockout, we had also a shift towards more advanced myelofibrosis. The changes in the compartments were the megakaryocytes were increased in the STAT3 knockout mice, and in particular, the megakaryocyte progenitors in spleen were clearly much higher than in the presence of STAT3. The target genes were affected by the absence of STAT3, and interestingly, one of the genes that was upregulated was STAT1 between here presence of STAT3, absence of STAT3. So it seems that as a compensatory mechanism, STAT3 is slightly upregulated. And that suggests that the effect on platelets that we see may be mediated through STAT1. And here we try to reproduce the data with a STAT3 inhibitor. You can see that the STAT3 inhibitor does have a slight effect on platelets here in the presence of STAT1, but when STAT1 is absent, this increase is abolished. So the STAT3 inhibitor in the absence of STAT1 cannot increase platelet count, suggesting that at least in part this effect is mediated through STAT1. So in summary, the STAT3 effects on erythropoiesis were minor or absent, so there was no major change in the erythropoiesis, but in the thrombopoiesis, the STAT3 normally has an inhibitory effect, and in the absence of STAT3, then there seems to be a shift towards more STAT1 activity, which then promotes megakaryopoiesis. I should emphasize that the STAT3 experiments were all done in transplanted mice where only the hematopoietic cells are deficient for STAT3. Ross Levine has published a paper where if you delete STAT3 in the microenvironment, you abolish a lot of the cytokine responses, and you do see a phenotype on ameliorating MPN. So these are the cell autonomous effects that are taking place inside the blood cells. So our STATs potential drug targets. STAT proteins act on DNA, so they're transcription factors. Here is in the middle you see DNA, and it's sort of facing out of the plane of the screen, and the STAT proteins dimerize, and they sort of like with two hands they hold this piece of DNA, and it is this red domain, the DNA binding domain, that then makes contact with DNA. This dimerization is dependent on these SH2 domains, which are again dependent on the phosphorylation of tyrosines that are mediated by JAK2. So the idea is to block this dimerization by preventing this interaction, and the development of STAT inhibitors was obviously slower than the one on the JAK kinases, but there are some promising new compounds that bind into this SH2 domain pocket and tend to prevent this dimerization and thereby the DNA binding. And here is one compound that was recently published by a Canadian group, which inhibited STAT5 in a nanomolar range and was selective against STAT3, so was predominantly concentrating on STAT5. So in summary, the STATs are potentially also targets for therapeutic intervention, and it is STAT5 which from these genetic studies would be the prime candidate, whereas STAT3 and STAT1 are mainly changing the phenotypic expression, but would be predicted to be unable to block development of MPN. So I'd like to stop here and show you the people in my lab who were involved in this work. Adrian Duick did the STAT1 analysis, and Jean Grisois the STAT3 knockouts. And this was done in collaboration with Stefan Dierenhofer, who did the pathology. So I thank you for your attention.